Happy morning, children. I feel extremely glad to welcome you all for this wonderful online learning. I'm glad that we have come to the end of this October month, and it's time for us to write our October monthly test. The portion for your October monthly test will be lesson 5 and 6. In our today's link, we'll be discussing about the 5th lesson question and answers. The answers are there and I'm going to revise those answers for you. Kindly watch it carefully. Mark the important key terms in your notebook. Learn those answers and revise them properly and get prepared for your forthcoming October monthly test. I wish you all to score more marks and be the toppers. Let's start with lesson 5, plant tissue culture. The first question, what is the name of the process given below? Write its 4 types. What is the name of the process given below? Write its 4 types. Okay. Now let's move to the answer. The process of plant tissue culture based on the explant is classified as organ culture, meristem culture, protoplast culture and cell culture. Okay, the process is called plant tissue culture. That is the first answer for the first question. The types organ culture, meristem culture, protoplast culture are the answers for the second one. What is the second question? Write its 4 types. Okay, you have to write 2 answers. Let's move to the 12th question. How will you avoid the growing of microbes in nutrient medium during plant tissue culture? So it has got 2 questions. The first one is how will you avoid the growing of microbes? Okay. The second one, what are the techniques used to remove the microbes? So 2 questions are in question number 12. The microbial growth in culture medium can be overcome by autoclaving the medium. Okay, the very important term is autoclaving. At what temperature? At 121 degrees Celsius for 15 to 30 minutes. So this is how you can remove the growth of the microorganism. And what is the process called as? It is called as chemical sterilization or simply sterilization. Okay, so chemical sterilization is using chemicals. Then the other sterilization using UV radiation. Alcoholic sterilization using alcohol, autoclaving and filtration. So these are the various techniques, but all these techniques together we call it as sterilization. So sterilization is the process for removing the microbes. How do you remove? By autoclaving it to 120 degrees centigrade for 15 to 30 minutes. Okay, now let's move to the 13th question. That is, write the various steps involved in the cell suspension culture. Write the various steps involved in cell suspension culture. Okay, we can find 4 points here. First is growing of the cell. Calyx in the medium, growing of the cell in the medium, in a nutrient medium. Second, transfer of the calyx to a liquid medium. We have to transfer it to a liquid medium. Agitation of calyx using rotary shaker. Put it in the shaker and you have to shake it well. Then filtering and separation of the cell. So these are the 4 steps which are used for cell suspension culture. 14th question. What do you mean by embryoids? Write its application. Very important question. What do you mean by embryoid? Okay. So here the embryoid is also known as somatic embryogenesis. What is embryoid known as? Somatic embryogenesis. It is the formation of embryo from the calyx tissue directly. Okay, and these embryos are called the embryoids. Usually embryoids are formed after the fertilization process, but here embryos are formed directly from the calyx tissue, and such embryos are called the embryoids. Okay, is that clear? or from in vitro cells directly form pre-embryoid cells which are differentiated into embryoid. In the in vitro, actually calyx is in vitro, inside the lab, okay. So embryo formed directly from the calyx are called the embryoids. Application, you have got some 3 applications. We'll go one by one. Somatic embryogenesis provides potential plantlets which after hardening period can establish into plants. So these embryoids have the ability to grow into a plants, okay. The second one, somatic embryos can be used for the production of synthetic seeds. First is they have the ability to grow into a plant. That is the first point. Second is they have the ability for production of synthetic seeds. Let's move to third point. Somatic embryogenesis now reported in many plants such as Allium sativum, Odium vulgare, Oryza sativa, Zea mays, and this is possible in any plant. It is easily seen in any plants, okay. So these are the plants which have made use of this process. So these are the applications of somatic embryo. Is it clear, children? So these are the 3 points you will find. So let me repeat. Calyx tissues directly give rise to an embryo which is called the somatic embryoids. In another word, in vitro, okay, in vitro cells directly form pre-embryoids which is called the embryoids. You got 3 importance. One is they have the ability to grow into a new plant, ability to grow the synthetic seeds, and they are commercially useful in production of certain plants. Now let's move to 15th question. Give the examples for micropropagation performed plants. Okay, it is plants. Micropropagation. The examples are pineapple, banana, strawberry and potatoes. Okay, pineapple, banana, strawberry and potato. Banana is also spelling mistake. Pineapple, banana, strawberry and potato. Is it clear, children? Now moving to 16th question. Explain the basic concepts involved in plant tissue culture. Okay, there are 4 basic concepts in plant tissue culture. They are totipotency, differentiation, de-differentiation, re-differentiation. Again let me say differentiation followed by de-differentiation, then with the re-differentiation. So 4 concepts are totipotency, differentiation, de-differentiation and re-differentiation. Let me start with totipotency. What is totipotency? The property of living plant cell that they have the genetic potential when cultured in nutrient medium to give rise to a complete individual plant. Okay, this plant cell have a property to grow into a new plant even when they are in the cultural medium. Okay, this plant cell have a genetic potential, they have the ability, they have the potential to grow into a new plant even when they are kept in the nutrient medium. So the property of a living plant cell that they have the genetic potential when cultured in a nutrient medium to give rise to a complete new individual is called the totipotency. Now let's move to the next topic, differentiation. What is differentiation? The process of biochemical and structural changes by which cells become specialized in form and function. Okay, the cells become specialized, they become differentiated. Before they are of same calyx. Now this calyx is undergoing changes biochemically and structurally by which the cell becomes specialized to form and function. So you can find the cell in the seed suddenly turns to plumal and radical. So plumal functions as the stem, radical functions as the root. So they change in their form and function. The third is redifferentiation. Okay, the further differentiation of already differentiated cell into another type of cells. Already it is differentiated. It is going to be further differentiated. For example, the component cell of calyx have the ability to form a whole plant in a nutrient medium. This phenomenon is called redifferentiation. Is it clear? The calyx, which again has got the ability to grow into a new plant, is called the redifferentiation. The fourth concept is dedifferentiation. The phenomenon of the revulsion of mature cells to the meristematic stage leading to the formation of calyx is called dedifferentiation. Okay, you have to find the key term reversion of the mature cell. The cell has undergone growth and it has matured and it has become permanent cell, but now this mature cell turns to meristematic cells, that is the cells which are in continuous state of cell division. So the reversion of the mature cell to the meristematic state leading to the formation of calyx is called the dedifferentiation. So these are the basic four concepts of plant tissue culture. The next 17th question. Based on the materials used, how will you classify the culture technology? Explain it. Okay. How you will classify the culture technology? Explain it. Let's look into the culture technology. First, organ culture, meristem culture, protoplast culture, cell culture. So these are the four types of culture: organ culture, meristem culture, protoplast culture, cell culture. How we are making up this? Organ culture is for embryos, anthers, root and shoots are used. In meristem culture, meristematic tissues are used. Protoplast culture, protoplast are used. Cell culture, single cells or aggregations of cell from the calyx are used. So these are the components. In detail you will find it in your textbook. You can learn that. Give an account on cryopreservation. Give an account on cryopreservation. This is a very important technology, children. Cryopreservation, also known as cryoconservation. It is a conservation, preserving it. It is a process by which protoplast, cells, tissues, organelles, organ, extracellular matrix, enzymes, any other biological materials are subject to preservation by cooling to very low temperature of -190 degrees Celsius using liquid nitrogen. At this extreme low temperature, any enzymatic or chemical activities of the biological material will be totally stopped, and this lead to the preservation of material in dormant state. Okay, we are taking the calyx and cooling it very low temperature of -196 degrees Celsius using liquid nitrogen. At this very low temperature, the enzymatic activities are completely stopped, and this we preserve the material. So this kind of preservation is called cryopreservation or cryoconservation. Usually we use to preserve protoplast, cells, tissues, organelles, organs, extracellular matrix, enzymes and any other biological matter. Is that clear? These are the substances which are preserved by -96 degrees centigrade using liquid nitrogen, where the enzymatic activities are stopped and the cells are preserved. So this is about the cryopreservation. Later, these materials can be activated by bringing them to the room temperature. We can bring back them to room temperature where all the enzymatic activities can start. How can we protect them? This has to be protected, okay, by adding dimethyl sulfoxide, glycerol or sucrose before cryopreservation. These protective agents are called the cryoprotectant. Okay, we are called cryoprotectant, dimethyl sulfoxide, glycerols or sucrose. They are used to protect them because these tissues, they undergo stress while freezing. We are putting it into temperature -121, 196 degrees centigrade. At this temperature, the cells will undergo stress. So to protect the cell, we are adding the cryoprotectant. Now let's move to the 19th question. Very important question. What do you know about germplasm conservation? Describe it. Previous one is cryopreservation. Now it is germplasm conservation. It refers to the conversion of living genetic resources like pollen, seeds or tissues of plant material maintained for the purpose of selective plant breeding, preservation in live condition and used for many research works. Okay, so we are conserving the living genetic resources like, okay, these are in live condition, okay, like pollens, seeds or tissues of plant material. For what purpose? For the research work. Okay, germplasm conservation resources is a part of collection of seeds and pollens that are stored in seed or pollen bank. Main aim is to conserve the seeds and the pollen. These two are stored in the seed or pollen bank so as to maintain their viability and fertility from any later use such as hybridization and crop improvement. Germplasm conservation may also involve a gene bank. It is involving with a gene bank. Now what is a DNA bank? Of elite hybrid lines of plant resources for the maintenance of biological diversity. We call these banks as the gene bank, DNA bank. Okay, we store these materials in the bank for our maintenance of biological diversity. Now let's move to the last question. Write the protocol for artificial seed preparation. Protocol. What are the steps involved in the artificial preparations of seed? Okay, first we have to development of calyx through PTC. It is nothing but plant tissue culture. Development of calyx through plant tissue culture. Second, induction of somatic embryogenesis. We have studied about somatic embryogenesis, that is development of embryo from the calyx. Maturations of embryoid. Then encapsulation of embryo. We are covering the embryo using agarose and sodium alginate. The embryo is formed, the embryo is matured. Before the embryo grow into a plant, we are covering, we call it as encapsulation. Encapsulation of embryo using the material. What are the two material? Agarose and sodium alginate. And this is called artificial seed. The other name for artificial seed is synthetic seed. Is it clear, children? So these are the steps. First we allow the calyx to grow in plant tissue culture. Then embryogenesis is formed. That is development of embryo from the calyx. The embryo is allowed to mature. The embryo is encapsulated by agarose and sodium alginate. And lastly, artificial seeds are produced. I believe these 10 answers will help you to revise all the questions of the lesson 5. Learn it thoroughly, prepare well and do your exams well. All the best, children. You can write a home test of these questions and post it in the group. Thank you, children.